tumultuous afternoon because you have talks upon talks without any break. I'm going to try really hard. First, I really want to thank Gadi for inviting me. And in case you haven't noticed, my international perspective is actually a black perspective. And that's really because, as Ken said, we've sort of been in the midst of trying to do this work together. And every time I really get to meetings and we're really talking about fantastic genetics that you can do in a homogeneous population, I'm really thinking about, wow, how challenging it's going to be to think about it in the rest of the world. And so my view and what I want to share with you is really sort of the rest of the world. And the rest of the world, the way I'm thinking about it these days, is that we've gone beyond single gene analysis to actually thinking about large genomic analysis. And that because we can't actually only think about BRCA1 without thinking about the tumors that develop in BRCA1 mutation carriers, I just want to put your head around this fundamental shift in our thinking about breast cancer. We thought it was one disease, and it wasn't really until we found BRCA1 and we could now classify the most aggressive type of breast cancer as the basal-like breast cancer that everyone really started getting scared of breast cancer. And we made the decision that there were some cancers that were sort of indolent because they were the individuals with lumina A breast cancer lived a little longer. Now remember that this is a short follow-up. We treat women with ER-positive breast cancer for 5 years. And that's really what's been the difference in terms of how we interpret genetic predisposition, the tumor you get, and the treatment you get. So the lumina A breast cancers are abundantly ER-positive, and for decades we've treated them with tamoxifen, and then we realized that the BRCA1 mutation carriers get triple-negative breast cancer, and that's why every oncologist has been really thinking about, let's get the breast off, let's get the ovaries off, and let's not risk having these women develop cancer at all. Well, now let's come back 20 years later. What has happened because we found BRCA1? Well, everyone is really quite excited, and you heard from Judy about the fact that platinum is clustered with, in the basal-like subgroup. It really got everybody's attention. So that's sort of an introduction to how I'm thinking about genomics. Genomics talks about ancestry, Ashkenazi Jewish ancestry. I'm thinking about sort of the world and all of our genes coming from African ancestry, and the fact that you have, at least based on new knowledge of population genetics, that there's really been a significant migration of people out of Africa. You have your Ethiopian Jews that are now here in Israel. You have individuals who came out of the west coast of Africa, came through the slave trade, and are in the Caribbean. And you have individuals sort of really all across the diaspora. Yes. So the question is, how small or how diverse or how great, what country you are in, there is certain fraction of their early onset breast cancer that have mutations that have founder effects. The reason why no country other than Iceland and maybe now with Steve's study in Poland have been able to use founder mutations for population screening is because many of these founder mutations that would have a founder effect will not be able to cover the entire population. And it's really not a wise strategy to use those founder or those recurrent mutations as a population screening strategy. So we've been doing work in Nigeria, and we of course, have never come across any 185 del right? However, anywhere there's the Jewish diaspora, you're going to find 185 del and a lot of the mutations that were previously thought to be only common among Jews. I practice in Chicago. I have more Polish women with Polish founder mutations in my practice than probably people who will practice in Warsaw. Because outside of Warsaw, Chicago has more Polish women than any other country in the world. The same thing, depending on where people have gone in the diaspora, these founder mutations actually impact what kinds of tumors they have. When I found my Hispanic patients with 185 del AG, the question I asked them is, are you Catholic or are you Jewish? Many of them are now Catholic, but we know that it's because they're part of the Jewish diaspora. So now let's think about how do we actually roll out these mutations and how do we use them in the clinic. I think one thing that's been really clear is that in the clinic, every oncologist wants to know what's the estrogen receptor, progesterone receptor, and HER2 for these tumors. And the reason they ask that is because they want to know what treatment to use. So a lot of population-based studies, and our study in Nigeria was actually the first to ask the question, when you walk in through a doctor's office, what is the diversity of tumors that comes into your clinic? Because in my clinic on the south side of Chicago, I see a lot of triple-negative breast cancer. And there may be some oncology practices where they never see any. And it's really because of the populations that come through my clinic. So when I started noticing that, and because of some of the work that's been done in Memorial and other Jewish hospitals, we went actually to Nigeria, and we wanted to look at, and the North Carolina study was also looking at the pattern of cancers that were coming in through the clinic. And you can see clearly here that the Lumina A breast cancer is actually the minority, if you have less Lumina A breast cancer in this, the more African ancestry you have. And you can see the Asian ancestry and white ancestry, you have a lot of Lumina A breast cancer. So what's driving that? Is it genetics or is it just lack of access and poverty? So a lot of people would really think about disparities in outcomes based on whether people have access to treatment or not. But I've actually been very interested in what are the genetic determinants of molecular subtypes, because we actually have a genomic determinant of it. So if you look at based on our studies, SIA now they're mandated in the US that you should do ERPR on all SIA database. And clearly, what you find is that depending on whether you're white or African American, you're more likely to have triple-negative breast cancer and HER2-positive breast cancer. If you're Asian, you're more likely to get HER2-positive breast cancer. There's some SNPs that actually predict these molecular subtypes, including a SNP that's very common among Asian populations. So your population genetics matter, and your genomics really is determined by your germline. So when then we ask the next question, what is the likelihood that you're going to die from breast cancer? So this is sort of the population genetics of how people get different breast cancers. And SIA would say, well, here's the incidence rate. There's no difference, white or black. There's a lot less breast cancer among Asian, Pacific, American, Indian, and Hispanics. But overall, if you look at non-Hispanic versus Hispanic, there's a lot more breast cancer. So then the question is, how about death rate? And you can see the reason why people talk about blacks dying at a higher rate than whites in the US is because of this staggering data. So then we ask the question, what is causing that mortality? Are there population differences in pathogenic mutations, and do they explain health disparities? To cut a long story short, the more we look for BRCA1 mutations, the more we find them in all populations, and the more we test more populations, the more we run into variants of unknown significance. And the reason why we have variants of unknown significance is because of the diversity that's in the African genome. So we've published about this classified variants. We were looking for recurrent mutations. We could never find them because, as I said, the diversity. So why so many variations? Because life began in Ile-Ife. How many of you know where Ile-Ife is? That's the center of the world in Yoruba Kingdom, right? And so when you're thinking about ancestral origin, when you're thinking about genetics, now why we're really asking that we go back and sort of get to the root of all diseases by mapping disease genes in Africa, it's really because of this population geneticists will tell you that out of Africa arose the entire human race. And as a result of that, that's why 99.9% of our genome is exactly the same. But that 0.1% that's different is being acted upon by different environmental factors, and genes have been selected for. Dr. Offit just told you that, of course, BRCA1 was not selected for, but there are many reasons why you have to actually look at this out of Africa pattern of migration. So we then ask the question, is the burden of early onset breast cancer going to be distributed based on the patterns of genes? And this will be rare alleles, right, of genes that are predisposed to adult onset cancer. So we then started doing a lot more BROCA analysis, which is a panel of genes in our African ancestry patients. And the reason we wanted to do that is because we want to annotate all of the variants that we're going to find. The bottom line is that if you look at BRCA1 and BRCA2 consecutive African Americans that we have, they have just as high a percentage with mutations as you see in Jewish women. The only difference is that because of the founder mutations, it's a lot easier, and because of the homogeneity that you see in Ashkenazi population. So 68 damaging mutations in 8 genes are identified in 65 patients. So those are the ones that we know are clearly damaging, but ask me about all the variants. We've done the analysis. It doesn't matter whether you get cancer early. If you get it early, you're more likely to have BRCA1 or mutation. But if you test anybody at any age, African ancestry, we find mutations in these genes. Then we done BROCA panel on our Nigerian cases. We even see more diverse variants and more diversity and no founder mutations because, of course, of the genetic diversity in the population. However, they still have predominantly BRCA1 and BRCA2, just like Ashkenazi Jewish populations. When you look at cases and controls, and we're continuing now because we really want to know how these variants are segregated across cases and controls. You had 3 mutations that explain 98% of breast cancer and ovarian cancer among Ashkenazi population. Look at the spectrum of mutations. It's like everyone has their own personal BRCA1 or BRCA2 mutation.
3 minutes.
So, 3 minutes. So we've done this study. We see these mutations. They're common. The variants are there. And the important thing is, if you have a mutation in BRCA1 or BRCA2, you are 20 times more likely to get breast cancer than if you didn't, thinking about cases and control. So if you can identify everyone who has 20 times more likely chance of getting breast cancer, wouldn't we all want to do that? So one of the things that's also very important is sort of work that's coming out of the International Cancer Genome Consortium, where people are actually looking at geography and your mutational signature. Because it turns out that, in fact, the pathway to progression may be different depending on your germline genomics. The Cancer Genome Atlas is also thinking about all of this stuff. And I just want to end by showing you that not only are we talking about single genes, we know that there are variants in microRNA, that the proteins that are made are going to be dependent on the interactions between your mRNA, your microRNA. We know that the DNA copy number or the African or even any genomic structure that you're going to start off with is going to be different. The patterns of methylation is going to be different based on your geography and where you live. And this is why the challenge for us geneticists who are actually trying to think about penetrance, who are trying to advise family members, becomes even more complicated because you are much more than your genes. So in the Cancer Genome Atlas, we looked at individuals who reported as being Asian, Caucasian, or African, or Black. And you can see, using principal component analysis, that if you're black and you know you're black, you're probably black. If you're in between and you're not sure, you're probably not sure. And so principal component analysis can actually sort you out one way or the other. However, if we use your ancestry to determine what kind of breast cancer do you have, this is work in progress. We find out that the more African ancestry you have, the more likely you're going to get HER2-enriched breast cancer and basal-like breast cancer. And that's really based on your germline genetics. And heritability of subtypes is there. We don't know what fraction is due to your germline genetics. So the question is, why do founder mutations just sequence everybody like they're doing at Memorial? Right? So everyone will tell me that, well, it's too expensive to do it. And I say that's not my job, whether it's expensive or not. My job is to go where the science is. And the science is that the WHO criteria for population screening for genetic predisposition to diseases. Disease is an important public health burden. Risk of disease due to mutations in the screen genes is known, and effective interventions to reduce morbidity and mortality among genetically susceptible exist. So if the WHO says that's what we should be doing, then I think that's what we should be doing. The challenge is that we haven't done enough science. We haven't looked at the international diversity by geography and ethnicity. So I'm calling all of you in this room to really think about how you can contribute to Bridge. We have data from North Africa, Moroccan Jews. I mean, you have access to Arabs, and we need to populate this Bridge consortium because we need to look at mutation spectrum, cancer risk, and we don't need to only depend on Angelina Jolie as the advocates for individuals with inherited mutations that cause cancer. All of us should be advocating for that type of genetic justice, because we know that not all women need risk-reducing mastectomies, but BRCA1 mutation carriers need oophorectomy. Now, BRCA-associated cancers benefit from PARP inhibition, whether it's prostate, whether it's breast. So we need to advance that science, and we need to advance it all over the world. So my final thoughts are high burden of premenopausal breast cancer, and we need to accelerate progress in using genomics to improve health outcomes. Thank you.